Hi everyone. Today I will be talking about genetic recombination in coronaviruses, and specifically how genetic recombination found in coronaviruses can give rise to novel coronaviruses, such as the coronavirus that's causing the COVID-19 disease and the current pandemic that we are experiencing. So before I begin, I will just go through some background information that will help you guys to understand what I will be talking about later on. So there are 4 genuses of coronavirus. There is the alpha genus, the beta genus, the gamma genus, and the delta genus. The alpha and beta genus are mainly composed of mammal coronaviruses, whereas the gamma genus is composed of only avian coronaviruses, so coronaviruses that affect birds. Within the beta genus, there are 4 subgenuses called lineage A, lineage B, lineage C, and lineage D. So in this graphic, I put in some examples of human coronaviruses in gray. So all of these names in gray are coronaviruses that infect humans. So in the alpha genus, there are 2 coronaviruses that infect humans, 229E and NL63, and then all the other human coronaviruses are found in the beta genus. So within the subgenus lineage A, there is a human coronavirus OC43 and HKU1. And then in lineage B, you have the SARS coronavirus that caused the SARS outbreak in 2003. And then SARS coronavirus 2, which is the name of the coronavirus that is causing the current outbreak. In lineage C, you have MERS coronavirus, which caused the MERS outbreak in 2012. So over here to the right, I just have an image that I got off of a paper showing a phylogeny of all of the genuses of coronaviruses that I just talked about. So there is the alpha here, the beta genus here, the gamma, and then the delta genus, which are condensed in order to fit all of these names in. But you can also see within the beta genus how there's the A, B, C, and D subgenus. So a little bit about how the viruses spread from organism to organism is shown in this image. So first I'm just going to define reservoir, or a host reservoir or natural reservoir, and this just means the organism in which the virus normally lives and reproduces. And typically they will live and reproduce in this organism without harming the organism that it's in, or without affecting the organism that it's living in. An intermediate host is just an animal that can also be infected by the virus, but can spread it to further organisms, such as how the civet and the camel can spread it to humans. So in this image, the solid arrows represent confirmed modes of transmission, whereas the dashed arrows represent possible modes of transmission that have not been confirmed yet. And so something that is especially important is in this image is that these 4 coronaviruses, NL63, HKU1, OC43, 299E, they do not have a zoonotic reservoir. And what's common among them is that they are also less severe. So the symptoms of these coronaviruses include symptoms that are similar to having a common cold, so like having a cough, a fever, a sore throat, or a runny nose. And all of these coronaviruses can be spread from humans to other humans, and they're not maintained in a animal such as the MERS and SARS. And so the MERS and SARS, they both have zoonotic reservoirs, and they're also both a lot more deadly or pathogenic than these other 4 coronaviruses. And it's believed that bats are the host reservoirs for the MERS and SARS coronavirus. And the civet is believed to be the intermediate host for the SARS coronavirus, while camels are believed to be the intermediate host for the MERS coronavirus. It is very probable that genetic recombination occurred in the civet and the camel before the virus spread to humans. However, that is not a definite fact, I guess I could say. So here I'm just going to talk about the structure of coronaviruses. In this image, you can see a coronavirus cut in half. The yellow is the spike glycoprotein, and then in this pink spiral is the single-stranded RNA that all coronaviruses have, and the nucleocapsid protein. You have in red the envelope, and then in blue the hemagglutinin esterase dimer. So what is especially important in this image is the spike glycoprotein. And this is because the spike glycoprotein is the protein that allows the virus to bind to the host cell to infect it. And within the spike glycoprotein, there is the receptor binding domain. So in white are the receptor binding domains. And the receptor binding domains are what is specifically from the spike protein, what is responsible for recognizing and binding to the cell surface receptor or enzyme of the cell that it's infecting to allow it to fuse with the cell and transport its viral genetic material inside. And this is important because it needs to transport its viral genetic inside of a cell such as a human cell, because it uses the machinery in the human cell to replicate its own genome to create more viruses. So here's a ribbon model of the SARS coronavirus 2 spike, and one more time, the SARS coronavirus 2 is the one that's causing the COVID-19 disease that we're seeing right now. And its receptor is the ACE2 protein found on human cells. So what it will do is it will use its receptor binding domains to bind to the ACE2 protein on human cells, and then it can infect the cell and replicate. So recombination can happen among different strains of the same coronavirus or between coronaviruses belonging to different genuses or subgenuses. And there are many factors that increase the chances of recombination occurring in coronaviruses, one of which is how coronaviruses have very high mutation rates because their replication fidelity is actually quite low. Their replicase makes a lot of mistakes when it is replicating its own genome. And so this can drive viral evolution and genome variability. Another reason why recombination occurs often is because coronaviruses create subgenomic RNAs. So this image is just a demonstration of what subgenomic RNA is. So if you have your original template strand, here, a subgenomic RNA just means that the replicase will start replicating the original template at the 3' end, but oftentimes it will jump to some point closer to the 5' end. And this point that they jump to can be different in all of the templates that they make. And so what happens is you'll have a lot of templates with gaps in between, but these templates will have sections of it that are similar to each other. And so the similar regions can allow for genetic recombination to occur. Additionally, coronaviruses that can be found within multiple host species can increase the rate of recombination events because there's a greater chance of a recombination event to occur when coronaviruses are found in more hosts. Because there are so many different types of coronaviruses found in all these organisms, genetic recombination between these different coronaviruses will occur if they are both infecting the same organism. The importance of recombination is that a lot of times recombination will enable a virus to escape host immunity or to develop drug resistance, which will allow it to live longer and replicate and produce more offspring, I guess you could say. So recombination is a very common way for novel coronaviruses to form. And oftentimes it is in the spike protein, because if you have recombination occurring in the spike protein, this will alter its surface antigens that immune system cells use to identify the virus and to kill it. But if you have recombination occurring and create a spike protein that is slightly different, this will allow the virus to escape the immunosurveillance in animals so that it can adapt to a different host, such as a human host. And so because of this, the S protein is often a hotspot for recombination studies. However, there are some requirements in order to produce a viable novel coronavirus. So obviously you need two coronaviruses to infect a host at the same time, but they also have to continue to replicate without interfering with each other. There also needs to be enough similarity between their genome sequences to even allow recombination to occur during RNA replication. The proteins that arise from the recombination event also have to be functional. And lastly, the recombinant virus must have some selective advantage in order to survive. So while recombination does happen quite a lot, the generation of novel coronaviruses is actually quite rare. And so in this image, you can see a bunch of different types of coronaviruses and their genome shown in a block format. And everywhere that is colored represents a recombination event that occurred to form its genome. So you can see how almost all of these coronaviruses had a recombination event to form its genome. Down here you can see the SARS coronavirus, which has recombination with a lot of different other coronaviruses. Many of the molecular evolutionary methods to detect recombination events involve the analysis of multiple DNA sequence alignments. And one thing I'll talk about later on is that if the phylogenetic tree of a non-recombinant region, so say a region here that is white, if the phylogenetic tree of that region is different than the phylogenetic tree of a recombinant region, you would know that recombination has occurred. And this is because if recombination hadn't occurred, the phylogenetic tree of the two regions should be the exact same. You can also take a phylogenetic tree of the whole genome and compare it to the phylogenetic tree of a recombinant region, and that would also give an indication of if recombination occurred or not. So I'll just go over a case study of the SARS coronavirus, just because it's very similar, the methods that many researchers have used to detect recombination. It's very similar to how they are detecting recombination in the SARS coronavirus too. So in one of the papers that I read, what they did was they took DNA segments and aligned them using a recombination detection program called Plato. And this program detected any segments that had possible recombination. So what they did was they made a phylogeny of all of the DNA segments as well as of two different segments. And here you can see their results in this image. So this first image is the phylogeny that was formed when they created it using the whole genome. So you can see the SARS coronavirus, TOR2 is just a specific SARS coronavirus that they used. You can see how it is most likely grouped with beta coronaviruses. So group 2 is also known as beta coronaviruses. However, in B and C, they use different segments which the recombination detection program Plato indicated that it was a recombinant region, and they made a phylogenetic tree with those regions as well. When they made a phylogeny with these recombinant regions, the SARS coronavirus was grouped with another genus. So in this image here, the SARS coronavirus was actually grouped with avian coronaviruses, whereas in this image here, which they used a different segment of the DNA to create this phylogeny, the SARS coronavirus was actually grouped with alpha coronaviruses. And so this is really good evidence that recombination had occurred. The regions that actually had recombination occurring was the spike protein and the RNA-dependent RNA replicase. So this image here, as well as this image here, which is just another way to view the phylogenies of the non-recombinant regions versus the recombination regions. And again, you can see how the SARS coronavirus is grouped or most closely related to a different coronavirus in each of these phylogenies. So because of this, it suggests that SARS coronavirus was recombined with a member of the gamma genus and the alpha genus. And it's most likely an ancient occurrence because these branches here represent time. And so you can see how the SARS coronavirus branches away from it quite far back, and it's not a recent occurrence, because if it was recent, you would see that branching occurring somewhere here. So now that I have talked about the SARS coronavirus as kind of a case study, I will talk about the SARS coronavirus 2, which is what is currently causing our pandemic. So I decided to make a phylogenetic tree myself with the SARS coronavirus 2 sequence. So I used whole genome sequences, and I used MegaX software, which allows you to align sequences and make trees. And I aligned the sequences using the Cluster W method, and the tree was made using the maximum likelihood method, and I just kept all the parameters as the default state. Here's the accession numbers for GenBank in case you want to look up the sequences yourself. But here's a tree that was formed, and you can see that the SARS coronavirus 2 is most closely related to a bat coronavirus, RATG13, which is just the specific name for that coronavirus. If you go further back, you can see that the next most related coronavirus is the pangolin coronavirus. And here, the human coronavirus 229E was just used as an outgroup because it is in the alpha genus, whereas these four are all in the beta genus. However, what's interesting is that a paper that came out several days ago actually found that a specific region of the SARS coronavirus 2 genome is actually most closely related to the pangolin coronavirus instead of the bat coronavirus. And so this image here is a similarity plot of the SARS coronavirus 2 to the bat coronavirus and the pangolin coronavirus. So in green is the bat coronavirus, in blue is the SARS coronavirus. Both the green and the blue are bat coronaviruses. These are just two different specific types of the bat coronavirus. And then in red is the pangolin SARS coronavirus. And so in this region in orange, you can see how the pangolin coronavirus is actually most similar to the SARS coronavirus 2, whereas in all the other regions around it, it is the bat coronavirus. And this region is coding for the receptor binding domain region of the spike protein. And so what is believed because of this information is that a recombination event occurred with a pangolin coronavirus in the receptor binding domain region of the genome, which would explain why this region is more closely related to the pangolin coronavirus rather than the bat coronavirus. And so this image comes from another paper. This is the phylogenetic tree, a very simple phylogenetic tree. When you make it with the whole genome, you can see that indeed the SARS coronavirus 2 is most closely related to the bat coronavirus. Whereas if you create a phylogenetic tree with the 5 key amino acids for the binding to ACE2, and these 5 key amino acids are in the receptor binding domain of the spike protein. If you make a phylogenetic tree with this information, you will see the human coronavirus is actually more similar to the pangolin coronavirus. In fact, these 5 key amino acids are identical, whereas the bat coronavirus has 4 different amino acids when compared to the human SARS coronavirus 2. So, in conclusion, it is possible that the pangolin was an intermediate host for the SARS coronavirus 2. This image shows how bats and rodents are the natural reservoirs of a lot of the human coronaviruses, and then how the SARS coronavirus was passed to the civet, which was the intermediate host, which passed it on to humans. MERS coronavirus was passed to camels, which was an intermediate host, which then passed it to humans. And for SARS coronavirus 2, it is still being tested and it's not confirmed yet, but it is most likely the pangolin that was the intermediate host that then passed it to humans. And this is because of the receptor binding domain region of the genome that I just talked about, where that region specifically was more similar to the pangolin coronavirus rather than the bat coronavirus. However, further research will be needed to elucidate the exact evolutionary pathway of the SARS coronavirus 2 in animals. And here are my sources for this video. Thank you for watching, and I hope you enjoyed it.